Hello, everybody, and welcome to this morning or afternoon or evening section of the BGRS conference, depending on where you are currently located. It is evening in Novosibirsk, and it is afternoon or actually late morning in Moscow, where I am. So the section is on genomics, transcriptomics, and bioinformatics. It will take two halves, and I will chair the first half. My name is Georgi Bazikin. First talk today, so it will be rather diverse, as you see in the subjects covered. The first half will be almost solely devoted to quantitative genetics, and the first sort of introductory talk on that will be by Yuri Aulchenko. Yuri, you have 30 minutes, and there will be time for questions after that.
Dear colleagues, it's a real pleasure to open this afternoon session of the BGRS 2020 Symposium on Bioinformatics, Genomics and Transcriptomics with a talk on uses of quantitative genetics and genomics as a tool to study human biology. I would like to start with reminding that in life science we are dealing with extremely complicated, complex systems. What scientists normally do when they deal with very complex systems are the experiments. An experimenter would take a very complex system and he would try to change something to perturb this system. And then he observes what are the consequences of the change. By conducting a series of experiments, it is possible to hypothesize the internal making of the system and how the system functions. Those hypotheses inform next experiments, and so the scientific progress goes on and on. But it is really difficult to study human biology. While we do have model organisms and in vitro models, in vivo, we are restricted to observational studies and very limited intervention. And here, genetics can help. One can see human genetic studies as a way to piggyback on the experiments of nature. What I mean is, of course, the fact that mutational process introduces many mutations in a genome of every human newborn. Due to the process of genetic drift and selection, some of these variants become rather common in our populations. For example, if we were to compare my genome to your genome, we will see difference in millions of sites. And from this genetic diversity, as well as from the diversity of environments, the human diversity arises. So the idea of using genetics to study human biology in vivo is to follow on the experiments of nature. It is to track what are the consequences of perturbing this or that DNA sequence in our genomes. Now, the beauty of genetics is that to apply to study any phenotype, any trait of interest, you should be able to do only two things. Firstly, you need to be able to measure genomes. Well, actually, this is what we are really good at nowadays. We can measure millions of polymorphic locations in thousands and hundreds of thousands of samples. We can now read full genomes using short reads or even long reads. So what's the second component to use of genetics as a tool to study human biology? The only thing you will need extra is to be able to measure the trait of your interest. It doesn't matter how complex your phenotype is or how much you know, as soon as you can technically measure it, you can start using genetics to study it. What you will do, you would be scanning through the genome and trying to detect if certain DNA variants relate to the phenotype of your study. Starting from these variants, you hope to learn which genes, proteins, pathways, and processes are involved. The method that is most commonly used nowadays to study human quantitative traits and multifactorial disease is genome-wide association study, or GWAS for short. How do you do this? Mind that you already can read genomes and you can measure the phenotype of interest. Let us assume that you study height, and let us look into a specific location in the genome where in the population two alleles, and let us call them A and B, segregate. Hence, you can see people of one of the three possible genotypes. So you go to a population and you sample a person and you measure the genotype and the phenotype. So here is this person, and this person is rather tall, and it's homozygous for the A allele of the polymorphism you are currently looking at. Okay, one data point is not enough to do anything, so you go out and you sample second person. And what you see is that the second person is slightly shorter than the first one, and it is heterozygous. But two samples are not enough either to do any statistics. So you go and sample next person, and next until your sample is large enough. And by large enough, we mean thousands of people. Eventually, you achieve a satisfactory sample size, and you start looking into statistical strength of the relation between the genotype and the phenotype. For example, you can use linear regression to estimate the coefficient of regression and significance of its deviation from zero. To characterize the statistical strength, you would normally take a negative base 10 logarithm of the p-value, and what you do next, you put this dot on a graph, and on this graph, on the y-axis, is the strength of the statistical significance, and on the x is the location of the polymorphism which you've just been studying in the human genome. But wait, you did measure whole genomes of the participants, so you are going to have millions of dots on this graph, which is often called a Manhattan plot. Millions of dots means millions of tests, so you need to be really careful what you call significant. Normally, we use a threshold of p-values less than 5 by 10 to the power of minus 8 to say that something is genome-wide significant in such experiments. So only the dots which pass this threshold will be called significant. Moreover, we also prefer the significant associations to be replicated, that is, to be experiment-wise significant and to show consistent effects in an independent set of samples. Okay, so far so good, but what do you get from a GWAS in terms of better understanding biology? Let us look into a very specific GWAS that was done to study genetics of lipid levels. You can see two strong signals on chromosome 19. Let us have a closer look at the one at the short arm. So we zoom in the region, and we can see that the strongest signal comes from a region upstream of the LDLR gene. This gene encodes for a receptor of low-density lipoproteins. It kind of makes sense. So is this how we use genetics to understand human biology? Do we just look at the GWAS hits and the closest gene is the one we should implicate? Often, the answer is yes, indeed. However, as many geneticists will immediately guess, the answer is often not so simple. To give you an example, let us look into another phenotype, obesity. Obesity is influenced by environment, but it's also influenced in large part by genes. We can measure obesity, for example, by computing body mass index. We can measure it. Hence, we can use genetics to try to understand its biology. Very large GWASs were done for body mass index. Here I show you the results from a study of more than 300,000 individuals published in Nature in 2015. You can see that there is a very strong signal on chromosome 16. Actually, it was detected as early as in 2007 during the very first wave of genome-wide association studies. Let us look closer into this signal. It's nicely restricted to an intron of the gene called FTO. If you look on the internet what FTO stands for, it stands for a fat mass and obesity associated gene. Looks like an obvious candidate, right? Wrong. This gene was named this way exactly because in humans we found an association of polymorphisms aligning its intron with fat mass and obesity. What it codes for is an alpha-ketoglutarate-dependent dioxygenase, and linking this enzymatic activity to obesity is not trivial, although many people did come up with ideas how this may work. The trouble was always that experiments in model organisms and in cell lines were quite inconclusive, and for years people couldn't work out the exact mechanism linking FTO product to obesity. Now, many geneticists may ask, why would you concentrate only on FTO? Because we know that a functional element sitting in an intron of one gene may well regulate another gene. And this indeed has turned out to be the case. Using functional genomic data, both from public resources as well as those from they have generated by themselves, researchers propose that an intron of FTO contains a repressor. The obesity-associated sequence variant disturbs it and leads to derepression of two homeobox genes, RX3, which you can see here, and RX5, which is further to the right. So these genes are located at a megabase distance from the regulatory sequence that sits in the intron of FTO gene. It has been demonstrated that an increased expression of RX3 and RX5 leads to development of adipocytes with decreased mitochondrial thermogenesis, thus leading to overall decrease in basal metabolism. The hypothesis was rather conclusively proven in mouse gene manipulation experiments as well as in CRISPR-Cas9 experiments in human primary adipocytes. This is a rather exciting story. It took 7 years to understand how one very important locus works. Now, let us step back and try to understand what we have learned from these examples. So, here are GWAS results, and they link genetic variant to the phenotype. However, by itself, GWAS doesn't tell you about biology. The biology lies in understanding how the changes in DNA sequence translate into the phenotype, and this is not immediately obvious from the millions of dots we see on a Manhattan plot. A classical functional follow-up is a very long and labor-intensive procedure. At the same time, GWAS is a high-throughput way to identify loci, and we have thousands of loci identified, and for most of them, we do not quite understand the biology. And we cannot follow them using classical approach. What can we do? Well, what we should do is probably try to understand the consequences of genetic variation at many levels, from expression across tissues and developmental stages to changes in proteome and metabolome to cellular phenotypes and tissue function to finally complex traits. We have very large biobanks and cohorts to relate genetic variation to complex traits, to multifactorial disease. In order to track functional consequences of this genetic variation, we also need studies with very deep molecular phenotyping. And this is what started happening about 10 years ago. Now we have a range of resources that allows us to track functional consequences of genetic variation, and this helps us build in functional hypotheses in a way that later we can do very targeted experiments. Thus, when you'd like to learn human biology using genetics, if your phenotype of interest is complex, a good starting point may be to perform a large and powerful genome-wide association study. A great thing about a study of common standing genetic variation is that it is able to bridge the knowledge obtained on different data sets. This means that you can trace functional consequences of a variant that you have implicated in one set of samples in another set of samples. A set of samples that was phenotyped very deeply and that addresses very fine aspects of human functional genomics. At that moment, you will really need big genetic and genomic data, you will have them, and you will need to apply a wide range of bioinformatic and statistical genetic tools to build etiologic hypotheses, which then could be verified or disproved by very targeted functional experiments. It is worth noting that while computations here in the middle can be relatively quick, the other two elements may be still rather time-consuming. Of course, collecting phenotyping and genotyping thousands of samples takes many years, but even putting together a large consortium to establish GWAS may take a few years as well. You can also imagine that the last part of this journey, wet lab experiments, are rather time-consuming. So I have now presented a general vision on how quantitative genetics and computational functional genomics can be used as a tool to study human biology in vivo. In the few minutes that are left, I would like to tell you how my group practices this vision. As I mentioned, we see analysis of big genetic and genomic data as a very central thing to our approach, as a link between statistical genetics and biology, something that drives generation of etiologic and functional hypotheses. In about 2016, my group started developing the GWAS-MAP platform for aggregation, quality control, storage, and analysis of the summary statistics coming from genome-wide association studies. Since that time, we have also collected a very large database that contains more than 70 billion of genotype-phenotype association across multiple domains, from transcriptomics to complex traits. Those of you who are interested to know more about GWAS-MAP can attend the presentation of Tatiana Shashkova at the very end of this day. As I mentioned, a full cycle from GWAS to functional validation takes many years, and I am very proud that this year we have published a paper describing our effort that has started 7 years ago in 2013. In that work, we studied genetic regulation of glycosylation of immunoglobulin G. Immunoglobulin G is a very important antibody, and changes to its glycosylation tune its biological function. So firstly, we performed a very large GWAS. Many thousands of samples were used in the discovery stage, and many thousands of samples were used for replication because we really wanted to be sure about locations we discover. Secondly, we used obtained results and computational tools to postulate a gene network regulating IgG glycosylation. Finally, we performed in vitro experiments to confirm part of the postulated network. Lucia Claridge is going to describe this work in more details later in this session. At the moment, we are in the middle of a large project that will similarly investigate the biology of glycosylation of human plasma proteome. Currently, we have coordinated a consortium that jointly has access to phenotypes and genotypes of more than 10,000 people. This dataset is several times larger than any plasma proteome glycosylation dataset analyzed before. This work will be presented by Sotbor Sharapov later in this section. Another very interesting phenotype we are investigating is back pain. In a series of works, we have performed the largest genome-wide association studies and identified a number of loci related to this phenotype. The study of pleiotropy, genetic correlations, and pathway analysis suggests at least two strong molecular axes of back pain genesis, one related to structural and anatomic factors, and another related to the psychological component of pain perception and pain processing. Next, we performed the first ever Mendelian randomization study of body mass index and back pain. Mendelian randomization is a technique that exploits unique features of genetics to obtain reliable estimates of effects and to hypothesize on causal relations between phenotypes. In our study, we found a significant causal effect of body mass index on both back pain and chronic back pain. Finally, we started looking into a more broad phenotype of chronic musculoskeletal pain. We demonstrated that chronic musculoskeletal pain at different locations has a common genetic component. We suggested that this component is likely related to pain perception and processing. It should be noted that we also learned that the phenotype of chronic musculoskeletal pain is very complex, very polygenic, and we are not there yet to formulate new etiologic hypotheses and to start functional experiments. This exciting series of works will be presented in more details by Yakov Tsepilov, who will talk just after me. I hope that I have convinced you that genetics is a great tool to study human biology. Genetics can bridge disparate data sets, it can serve as an anchor for the array of causality, and it allows us to piggyback on the experiments of nature. With this, my acknowledgements firstly go to the great team that I am very happy to lead and to the collaborators that I am very happy to work with. I acknowledge support from the Institute of the Cytology and Genetics of the Siberian branch of the Russian Academy of Science and the Novosibirsk State University. I also would like to acknowledge support from the agencies that has funded the work. Finally, I would like to thank you for your attention.
Here.
Thank you very much for this broad overview, Yuri. And I believe we have plenty of time for questions. Thank you very much for this broad overview, Yuri, and we have plenty of time for questions. So I understand that the questions can be asked through the chat, and let me start. First of all, I would like to see to hear what is your perspective on the GWAS versus whole genome association studies done by sequencing versus done by whole genome sequencing or done by whole exome sequencing, the newer technologies, because GWAS has been around for quite some time. Are those newer things supposed to replace them, or GWAS is sufficient for all reasonable purposes? What is your feeling about that?
Well, I think that, well, firstly, I don't see much of the contradiction because I think many people contrast GWAS to sequencing-based studies, right? But I think this may be not entirely right. So what should be contrasted is array-based genotyping versus sequence-based genotyping, or you could call like whole genome sequencing. Maybe you shouldn't call it genotyping, but anyway, this is the way to measure your genomes, right? So there are like zillion of ways to measure genomes with different degree of precision, because if you think of this already, the knowledge that somebody is father of someone or mother of someone, this is the way to measure genomes, right? So it's like, yeah, if these are the first degree relatives, you expect or you even know that half of their genomes are the same. So this is like imprecise way to measure genome. And then, like in end of 70s, beginning of 80s, we could measure, few dozens and then few hundreds of RFLPs and then microsatellites and then arrays with hundreds of thousands or even millions polymorphic locations in the genome, and now we are to the sequencing. So like array-based versus sequencing-based. Sequencing is better. It gives you a more complete picture, especially when you think of low frequency variants, right? In respect of common variants, you're pretty well covered, at least in Europeans, with arrays. But yes, sequencing is better, and especially if you take long reads, then you have also these repetitive regions, which are very important for the immune system. So I'm all for whole genome sequencing with long reads, right? If you can afford it, because of course, array-based genotyping is, still several times less expensive. But then if you think, what is GWAS? GWAS is genome-wide association study. It's a kind of design to study complex traits, right? And it doesn't matter whether for genotyping you use array-based technology or sequencing technology. What may matter is that when you study rare variants, slightly different methods are being used. For example, you use the burden tests to bundle, rare variants and so on and so forth. But I wouldn't contrast. This is a very long way to answer your question. I'm sorry, maybe I'm talking too much, but coming back to this where I started, I wouldn't contrast genome-wide association studies to sequencing or array. I would say this is technology, and genome-wide association study is design. So there is no contradiction, and I'm all for genome-wide association studies by using sequencing. And we are going there. Actually, it's quite clear. It will be great.
I was not asking about the design. I was asking about the technology. So the question was, you have answered it sort of whether the technology is about to replace the chip-based sequencing.
Absolutely.
Okay. Okay then. So it looks like we do not have more questions so far. I encourage the audience to be more active in the hopefully in the next talks, which will be more specific and more detailed. And with that, I understand that we're taking we have 3 minutes till, or a couple of minutes till the next talk.